This video will be about synapsis of chromosomes during meiosis. Synapsis is defined as the pairing of two chromosomes that occurs during prophase 1 of meiosis, allowing homologous pairs to match up and a possible chromosomal crossover event to occur. Here is a quick illustration showing how homologous pairs match up during prophase 1 and synapsis occurs to create a crossover event. Let's check out an example with two individuals. Individual 1 has the following chromosomes. And if we flip the second one so we're reading from right to left, we can see that a pericentric inversion occurs with the four middle letters I, J, K, L. Put into an inversion loop, it will look like this, with the button representing the location of both of the centromeres. If a crossover event occurs between the J and K regions, it will form the following four chromatids. Starting with the top chromatid, we'll read it from left to right, writing down the letter of each gene loci as we pass it, including the centromere. Now we will follow the second chromatid, and this one was involved in the crossover, so we end up following the blue strand until we reach the crossover, and then we go back down with the yellow strand to the left, making sure to include the centromere. Now we will look at the third chromatid starting on the left. This one was not involved in the crossover, so we will write down the letters of each gene loci, including the centromere, of the yellow strand. Now we will look at the fourth chromatid starting on the right with the blue strand, and follow that until we reach the crossover, write down the centromere, and follow it back down with the yellow strand until we get to the end on the bottom right. Now we will look at each of the four chromatids and decide which ones are viable. Any chromatid that has a repeated gene loci or a deleted loci will not be viable, but the two that match the parent will both be viable. Now we will look at individual 2, who has the following chromosomes. And if we read the second one from right to left, we can see that no inversion occurs. In a crossover event between the J and K regions, the following four chromatids are produced. Starting with the top one, we'll read from left to right and write down each gene loci, including the centromere. This one is not involved in the crossover. Now we will do the second chromatid, again reading from left to right. This one is involved in the crossover, so we will follow the green strand until we reach the crossover event, and then follow the purple strand all the way to the right. We will look at the third strand that is also involved in the crossover event, and read it from left to right, starting with the purple strand, and then the green strand all the way to the right. Now we will look at the bottom strand and read it from left to right. This one is not involved in the crossover event. As you can see, even though there's a crossover event, there are four identical chromatids produced that match the parents. This means that there are four viable gametes produced. The inversion in individual 1 may lead to speciation, but individual 2 has the higher fitness, because of the fact that individual 2 produces 4 viable gametes while individual 1 only produces 2.